The first talk today is going to be by Dr. Kara Tsang, who is currently a postdoc in Professor Kat Holt's lab at the London School of Hygiene and Tropical Medicine in the UK. They've recently finished a PhD in Andrew MacArthur's group at McMaster University, which is the group that kind of runs and keeps active the CARD database, or the Comprehensive Antibiotic Resistance Database, which is one of the many AMR databases that Kara is going to kind of give a brief overview for, explain, what databases and resources are out there for AMR genomics. So if you're doing your own projects, if you're doing your own analyses of samples, what resources are out there to help you. So take it away, Kara.
Perfect. Thank you so much for the introduction, Finley. I'm just going to share my slides. Okay, so I'm very excited to be here and give an introduction of the databases and resources for AMR Genomics. So let's get started first with a brief overview about the purpose of these databases and resources. So as we all know, antimicrobial resistance, or AMR, is a global health crisis that is caused by microbes being able to evade the antimicrobials that we use in humans, animals, and the environment. So in this AMR workshop, I'll be focusing predominantly on bacteria, and they have many mechanisms through which they are able to resist antibiotics. So each of these diagrams represent a bacterial cell, and in blue is where they are susceptible through a particular mechanism, and in orange is where they are resistant. So I won't go through each of these mechanisms, but this is a good visual summary of all of the mechanisms, and it's important to understand the context and what we are actually trying to find when we talk about AMR genomics. So it's also important to note that these mechanisms can be categorized as intrinsic or acquired. So intrinsic resistance is defined as a naturally occurring phenomenon that predates antibiotic therapy and is present in all bacterial species. And a good example of these are the efflux pumps. Acquired resistance, on the other hand, is any mechanism that can be gained by susceptible bacteria. So each of these mechanisms can essentially be encoded in the bacterial genome through horizontal gene transfer, gene duplication, new promoters, point mutations, insertions, or deletions. And so these are the things that we're looking for within the bacterial genomes. And so in terms of bacterial genomics, after sequencing, we typically get sequencing reads. And if we're trying to find, for example, this resistant determinant highlighted in black, what we can do is essentially estimate the coverage of these reads that map to a particular resistance determinant that we're interested in so that we can identify it. With the reads, we can also assemble them into assembled contigs, which is representative of a bacterial genome. And so with these assembled contigs, what we can then do is identify these resistance determinants highlighted in black again through annotation methods that use alignment. And so these resistance determinants in black can represent any of these genetic features that I've highlighted here, and can only be found because researchers have discovered them, and we store these sequences in AMR databases. And so these databases can then in turn be used by tools to identify resistance determinants within your sequence of interest. So there are currently a number of AMR databases that exist and can be seen on this slide, some of which are actually not even listed here. And so more detail about each of them can be found in the review that I've listed in the citation. And so many of them differ in scope. So some of them can be focused on one very specific gene class. So for example, the beta-lactamase database focuses only on beta-lactamases, whereas some of them actually focus on a specific species. And in addition to that, some of these databases have associated tools that make annotating your sequence much easier. And others have not been updated for many years and are not currently active. And so because it would take hours to go through each and every single database, we have picked out 4 important databases and resources to highlight throughout this introductory talk. So the 4 that we will be going through are ResFinder, CARD, or the Comprehensive Antibiotic Resistance Database, the NCBI Resources, as well as the PATRIC Database. And so the reason that we picked these 4 databases is because they range largely in the species that they cover, the AMR families that they include, and the antibiotic resistance phenotypes that they focus on. All of these databases also rely on each other for information, and they do that, and they also provide their own expertise and uniqueness. Each of them also have custom annotation tools so that they specifically use their database and create their own tools so that it is ideal for the data that they have as well. Lastly, they are all regularly updated and very active in this field. And this last point is particularly important to emphasize because studying antimicrobial resistance is studying something that is like a moving target. Bacteria evolve very fast and way quicker than we do, and there are new mechanisms and genes that are discovered at a very rapid rate. So in addition to new mechanisms and genes, there are also variants of existing genes that are discovered, and they actually have sometimes very different phenotypes in the gene that it evolved from. And so it's important to keep track of all this information in the form of a database so that researchers can understand the current AMR context and maybe even use it to predict what's coming next. So AMR databases then really rely on published literature and domain knowledge experts to consistently add new findings. And sometimes it's not really just about adding new things, but it's also about removing or fixing mistakes in the databases to be reflective of what is currently known in the field. So anytime a change is made in a database, the versions of the databases changes incrementally. So versioning a database is important for reproducibility and to ensure consistent results across a study. So for example, if you were to use ResFinder version 1 versus ResFinder version 4.1, your results would be different, but the results from the current version would be more reflective of what is currently known about AMR. So now that we understand the genetic context and the importance of AMR databases, I'll give you a brief overview of these 4 popular databases and the resources that they also provide. So we'll start with ResFinder. So ResFinder focuses on finding acquired and/or chromosomal mutations in your sequence of choice, and they have specifically curated chromosomal mutations for these specific species listed here. And so what this means is that ResFinder does not include resistance determinants that are intrinsic, so this would include efflux pumps. And so this is the current distribution of their acquired resistance gene database categorized by drug class. And so we can see that a majority of the genes confer resistance to beta-lactams, aminoglycosides, and macrolides, and so on. The ResFinder database is divided into drug class. And so this is particularly useful if you're interested in studying a particular phenotype. And so, for example, if you were interested in rifampicin resistance, you could download this file and it would have all of the rifampicin resistance genes. And then on the right, we can see also any updates they have made to a particular database, anything they've added or removed or updated. In addition to this, they also have a step-by-step guide on their website about how to submit and interpret these results. And so I'll just give you a sneak peek here. So when you submit a DNA sequence to ResFinder, it will provide you with a phenotype result describing which antibiotic and antibiotic class that your sample is predicted to be resistant to, and then also provide gene descriptions for the reasons why. So for example, here, CTXM15 confers resistance to ceftriaxone, ceftazidime, and tigersilin. Below the phenotype results, you'll also get to see genotype results, and they're also organized by drug class. So here we have aminoglycoside resistance and beta-lactam resistance, and then the resistance genes that it has identified, along with corresponding information such as the percent identity of that gene that it found, the particular phenotypes, and any accession numbers and publications that you may be interested about this gene. And then again here, amphenicol resistance, but now these genes are highlighted in different colors because they are not 100% identical or do not cover the entire length of the reference sequence. And then below that, you will find any chromosomal mutations that were detected in a particular sample. And it makes sense that no chromosomal mutations were found because I actually input a Klebsiella plasmid into this particular example. And so next we will move forward and I will overview the Comprehensive Antibiotic Resistance Database, or CARD for short. So I will also mention that for a more detailed description of CARD, there are videos and tutorials in the GitHub of the website below. So CARD keeps up to date by comparing itself to other databases, looking at primary literature, and receiving knowledge, expert knowledge and feedback. And so because there is so much AMR information, not just the genes themselves that are important, there are many ways to store this abundant amount of information. So one useful way to do this is through an ontology. So unlike the other databases, CARD's underlying structure is an ontology, and specifically it is called the Antibiotic Resistance Ontology, or ARO for short. And so an ontology is just a network of words linked by relationships, and it is intended to connect knowledge. So this is a small example from the Antibiotic Resistance Ontology, and it is just showing how this gene, the immunocoumarin resistance GYR-B gene, is conferring resistance to novobiacin, chlorobiosin, and coumaricin here, and that it is also a type of immunocoumarin-resistant DNA topoisomerase. And so as you can see, this is sort of a network of knowledge that if you were to click on this gene, you would be able to access all of the things that it is related to. And so how this looks like on the website is if you were, for example, interested in the doxycycline antibiotic, you would click on this antibiotic and then you would see that it is within the tetracycline antibiotic drug class, and that it targets the 16S rRNA, and that these following tetracycline resistance genes confer resistance to doxycycline. And so even those who don't necessarily work with genomics or use CARD will use the ontology as a resource in studying AMR because it is a vast knowledge network. And so I've just briefly described CARD. It is structured as an ontology, and it also stores genomics reference sequences. So under each of the AMR determinants is a bioinformatics model that stores this genomic reference sequence. It is then used by the resistance gene identifier to predict resistance determinants within your sample of choice. And it categorizes each resistance determinant into different categories depending on how similar they are to the reference. And so I won't get into that tool too much, but I'll just highlight some other different resources that CARD offers. So here we have the CARD download page where you're able to download any relevant data files. So the ontology, the actual CARD data with the reference sequences. And then there's this useful file called CARD data changes, which can compare older versions to newer versions so that you can see what has been added if your results have changed from one day to the next. CARD also has a live dashboard that provides summaries of detected antimicrobial resistance for user-submitted genomic samples to the online RGI tool. And so this screenshot was just taken a couple of days ago. The CARD Live currently looks at 8,376 samples that were submitted online. Most of them came from Southern Asia, followed by North America, and then Eastern Asia. So overall RGI results from the CARD Live dashboard can also be categorized through drug class, AMR gene family, as well as resistance mechanisms through these very interactive plots. And so for time's sake, I'll just show the drug class resistance plots. And so here we can see of the percent of all of these samples, tetracycline, fluoroquinolone, and pan-am resistance were the most popular in these 8,000 samples. And then here is an upset plot of drug classes. And so you can see the most common intersections between the drug classes. And so just to give you an example here, there are 173 genomes that are both resistant to tetracyclines and fluoroquinolones, which is shown by this histogram plot here. And then the right-handed histogram shows the prevalence of each of the drug class phenotypes, which was shown on the previous slide. And then moving onwards, we also have a different resource called card prevalence. So the resistance gene identifier was used to analyze molecular sequence data available in NCBI genomes for 263 pathogens of interest, plus genomic islands that are available in the Island Viewer resource. And so this is just a screenshot of a part of the table that illustrates the species, the number of chromosomes, plasmids, whole genome shotguns, and genomic islands that we have run through RGI. And so you can also look at this from the perspective of the species. And so here we can see that, for example, 4.5% of the Acinetobacter johnsoni are resistant to acridine dye. And then if you wanted to also see this from the perspective of the gene, you can also see that, for example, here that 0.62% of the NCBI plasmids from this Enterobacter have this AAC3IB gene. So this is the card prevalence resource. And so then I'd like to highlight the card resistomes resource, which is a bit of a deeper dig into look at any of the particular genomes that you are interested in. And so, for example, here, if you were particularly interested in this plasmid from Staphylococcus aureus, you can see the resistance determinants that were identified within it using RGI, and then the drug classes that it confers resistance towards. And so at the gene level, you could also use the resistomes resource. And so if you were particularly interested in NDM4, you can see that it has been found in Enterobacter in the whole genome shotgun, Escherichia in plasmid and whole genome shotgun, as well as Klebsiella in the plasmid and whole genome shotguns as well. And this also speaks towards the ontology. If you're looking at this gene, you can see the different drug classes it's resistant to, the resistance mechanism, and any relevant publications that describe this gene as well. And so now I'll move forward from the CARD database to the NCBI resources. So as part of the National Action Plan, NCBI partnered with several outside agencies, including the FDA, the CDC, the World Health Organization, and others to take some of the following steps. So their first goal was to increase standardization. And so they've developed and maintained a curated database of AMR genes. Next, they wanted to make AMR-related data more widely available. So they collect genetic and antibiotic susceptibility data. And then lastly, they wanted to make more effective use of bacterial genomic data. And so NCBI has developed AMR Finder Plus to identify AMR genes in bacterial genomes. And so I'll just briefly touch on each of those resources today. And so this pie chart is just highlighting the distribution of the current organism phenotypes that are currently stored in NCBI. So as I previously mentioned, NCBI has a reference gene catalog that is also used by AMR Finder Plus to identify AMR determinants. And so NCBI has developed this database using a number of resources, including previous ones that I've mentioned in this talk, but I won't go through them all today, but it is just good to know that they are very thorough with their search. And so this is a screenshot of their database. And so you can change the number of columns that you would like to see. But for example, here you can see this is a particular gene name, and its product name, and then that it is an AMR gene, its class, subclass, and then any accession numbers, just in case you did want to get the sequences from RefSeq or GenBank as well. And so I'll just briefly mention the AMR Finder Plus tool that, in addition to identifying AMR genes, can also identify additional gene classes related to virulence and stress. Similar to ResFinder, there are also species-specific mutations that can be included or excluded for the species that are named in this table. And at the bottom, I've just linked to documentation on how to run AMR Finder Plus on the command line and how to interpret the results and more information about the reference gene catalog. So in addition to AMR Finder Plus, there is also the isolate browser that addresses clonality and describes AMR determinants in a given isolate. And so it integrates pathogen metadata, links to mutation clusters for genetically related isolates, and antimicrobial resistance genotypes and phenotypes, allowing users to browse and search over 300,000 pathogen isolates. So this is just a screenshot from the isolate browser, where we have a strain of Providencia. We have its identifiers, where it came from, the isolation source, and that it's a clinical sample, the antibiotic susceptibility testing phenotypes, as well as the resistance determinants that were identified using NCBI AMR Finder Plus. And then next, there is also a database for hidden Markov models, which is another way to identify resistance determinants that I won't get into detail today, but it is important to note that they have this. And then they also have the microbial browser for identification of genetic and genomic elements, or micro-BIG-E for short. And essentially every row in the micro-BIG-E display is an antimicrobial resistance stress response and or virulence gene that has been identified in an isolate with information about the method that was used to identify it, including supporting evidence, the element's type, subtype, class, and subclass, and additional columns if you choose to see them. So in NCBI, there's also the option to submit sequence and phenotype data so that your work can be part of their global collection as well. And so then now the last database that I'll go through is PATRIC, which stands for Pathosystems Resource Integration Center. And so this is an infographic that I've taken from their website, which describes where their data comes from and what services they offer. So at the genome level, they pull their genomes from the SRA, the ENA GenBank to get their genomes. And then any associated metadata comes from publications as well as biosample IDs. And then they generate what is called an AMR classifier, which is their method of identifying AMR determinants in a particular sequence. And then at the gene level, they curate their own database by looking through publications, using the CARD and the NCBI database, as well as perform some of their own manual curation to really define these AMR functional roles. And so then altogether, they provide this annotation service where they can annotate public genomes or private genomes that you choose not to share with the public. And so what they will provide you with is the predicted AMR phenotype. So the antibiotics that your sample is predicted to be resistant towards, any predicted AMR regions. So these might not be known AMR genes, but potentially new mechanisms or new resistance genes. And then lastly, they provide annotated AMR genes. And so these are the known AMR genes that they have curated in their database. And so here, I've just described the different databases that PATRIC uses to develop their own database. And so their database can be seen through their interactive table. And this is a screenshot of it. And so you can see the distribution of the sources that they use to create their database. And then every row essentially provides evidence for each resistance determinant. So here, for example, it comes from the literature and the source is CARD. And then it describes the AMR gene in more detail. And there's many more columns that you could look at if you wish to do so. Another resource that the PATRIC website provides is the AMR phenotypes for genomes. And so, for example, for each genome, it will provide the antibiotic susceptibility testing results and the methods used. So here we have this E. coli strain that was tested against meropenem, and it is susceptible, and the MIC is 32. And then if you scroll through the different columns, you'll also find the methods and the standards that were used for a particular strain. And then if you were interested in a particular genome, you could click on this genome, and then you would be able to see more information about it. So you can also see the MLST, for example, and also the full antibiotic susceptibility testing results here. And then if you scroll down, you can also see any metadata. So this isolate was taken from a slaughterhouse in 2015 from a cow. And then what we're interested in is the AMR annotation. So we can see the distribution of using the NCBI database, PATRIC, and CARD, and also different virulence factors as well if you were interested in that. And so you can also upload and annotate your own private data using PATRIC, and they have step-by-step tutorials on how to do so in this link below here. And so lastly, in addition to known AMR determinants, PATRIC also stores a database of computationally predicted small genomic regions that are associated with AMR. And so essentially, this leaves room for discovery and potentially new resistance genes and mechanisms, as these small genomic regions have not been experimentally validated to confer resistance to a particular antibiotic. And so this is just a screenshot of the PATRIC regions resource, where you have the genome name, the IDs, that it is a classifier prediction, where you can find it within a particular genome, and then what it is predicted to do. And so that is this is a summary of what I've discussed today, and this is just intended to be a refresher and a resource for you to look back at if you do choose to do some AMR genomic analyses in the future. And so I just wanted to end off this introduction to discuss about where we go from here. So there is a lot of standardization still to be done, and this is sort of on both sides. So standardizing the data before it enters the database, standardizing the storage, and then standardizing how it's outputted from the database as well. And so this is important because controlling and cleaning the information being put into the databases, controlling how it's used, and will really affect the downstream interpretation of the results as well. Even though there is some metadata that is included with the AMR databases, I think that there could be more. So for example, adding the elevation of minimum inhibitory concentrations in the literature with each AMR determinant would really help us understand the biological context of each of these AMR determinants. And my next point is that, as we have seen, ResFinder and NCBI AMR Finder Plus have both become species-specific with identifying chromosomal mutations. And so perhaps more species-oriented databases and tools are required for AMR genomics. For example, for something as complex as Mycobacterium tuberculosis, this could be useful. And then something that I'm sure the other speakers will touch on later is the need for the harmonization of all of the AMR databases, tools, and resources to be really able to compare and contrast what they are doing with the genomic data. And this in turn impacts our ability to interpret the results downstream. And so with that, I'd just like to thank the steering committee and the organizing committee for developing this workshop and making it so accessible. I believe my slides will be shared as a resource, and this talk is also recorded, but for now, I'd be happy to answer and take any questions that any of the participants have.
Perfect. Thank you so much, Dr. Tsang. That was excellent. So there has been a few questions, and a few that I know you have some good answers for and good opinions on. So one of the questions was, so when ResFinder or another tool reports a detection of antibiotic resistance genes such as SOL1, or if you're looking in the database and look up, say, SOL1, and you see it says, okay, sulfonamide antibiotic, does that mean it's resistant to all of the sulfonamide antibiotic classes? Is that relevant from a clinical perspective? How do you navigate the difference between antibiotic class level metadata and specific antibiotics across these databases?
Yeah, so that is an excellent question. And I think it depends on the antibiotic itself. But for example, something that I'm familiar with are the beta-lactamases. And so, for example, if I did find a CTXM beta-lactamase, I would not necessarily consider it to be resistant to all beta-lactam antibiotics. And because data has shown that specific mutations can actually change the phenotype of a particular CTXM beta-lactamase. And so I think that highlights a really great point that there needs to be even more curated data on that side in terms of not just labeling it conferring resistance to a particular drug class, but actually going down to that antibiotic level. And that is something that other databases such as CARD are trying to do to really get it to a granular level of which antibiotic does this resistance gene confer resistance to. It is just difficult because it requires experimental validation, and then you have to test all of the different beta-lactam antibiotics, for example. So it is a huge task effort, but it's definitely something that the field is striving towards for sure.
And I think you've raised in some of your previous work, one of the big challenges is actually the literature reporting these AMR genes. You have a lot of papers that, okay, we found a new AMR gene, but it's very inconsistent, the range of specific antibiotics they are actually phenotyped against.
Exactly, yes. They might test like one or two antibiotics and just leave the rest out, so you don't really know the full phenotype for a particular gene.
So I was wondering if you could talk a little bit about your microbial genomics paper, where you identified new substrate activity of known AMR genes.
Yeah, so in the microbial genomics paper, we essentially identified a bunch of different beta-lactamases, and then we noticed that in the literature, it wasn't really clear what phenotypes that they were actually producing. And so what we did was we used the antibiotic resistance platform, and we essentially expressed all of these beta-lactamases and tested a number of different beta-lactam antibiotics to show that these beta-lactamase genes actually confer resistance to different numbers of beta-lactams, and that wasn't previously reported in the literature. So we essentially showed new phenotypes for known AMR genes.
And as it says, this really does highlight that problem of not even beta-lactamases, which are relatively consistent. And I even see somebody further down was saying these databases seem overrepresented in beta-lactamases. Yes, they are. We have a lot more information about beta-lactamase-based resistance than a lot of other resistances in many ways, because it's very simple. It destroys the beta-lactam. It's easy, relatively easy compared to some of the more complex bits of resistance. And just inconsistencies in the way they are phenotyped in papers, especially including the beta-lactamase inhibitors and all the combinations of them. There's a lot of unnavigated space there. But I was wondering, especially as it's kind of coming up in a kind of theme in a few questions, and we'll probably talk about in the panel discussion later, but just about the general problem of going from genomes to AMR phenotypes and the real underlying challenge, no matter which database you use, no matter what data you create, the disjunction between genomic sequence and actual phenotypic resistance that you would be clinically relevant.
Yeah, so predicting phenotypes is a challenge from genomes. And part of the reason is that finding a resistance gene does not necessarily mean expression. And so that's one problem. And then another problem is that we don't know what we don't know. So there could be resistance genes that are in the genome that we don't know of, and so obviously we can't find them. And also there's a lot of other underlying factors about bacteria. For example, the environment they're in, if they're in a biofilm, that can affect phenotype as well. And so these sort of complicate things. And then at the species level also, something like E. coli is much easier to predict the phenotype compared to something like Pseudomonas aeruginosa. And then mechanistically, I think it's also important to think at the species level, because in some of my work, I've showed that, for example, for predicting phenotypes for Neisseria gonorrhea, for example, you have to work at the mutation level. And we know that antimicrobial resistance in Neisseria is driven by mutations, whereas something like E. coli and Klebsiella, it's much more of an acquired gene problem.
Yeah, and also I'd tie into that as well. It's just the way we measure resistance in a lab. Yeah. It's very different from the real world in a body situation, as I'm sure the more biological among the audience have a lot of unpleasant familiarity with. Okay, so here's again probably an item of general panel discussion, but okay, there's lots of databases. Which one should you use, and why shouldn't you just use one that combines all of the databases? So what might be some downsides of that?
Yeah, so I think you also have to look at, your own research question, right? You have to figure out like what species you're working with, what research questions you're working with, and which tool or database would fit the best. In an ideal world, of course, you could test all of them and, like collate all of those results and find something, but like that's just not time sensitive. And so one issue of sort of using a tool that, collaborates or uses all of them is that it may be difficult to like backtrack if like something goes wrong or like there's a result that you don't expect or et cetera, like backtracking to figure out, from which database it came from and how that has affected your ability to interpret your results.
Yeah, and as Kara says, one of the major questions I think you have to decide based on your own research is what is more important: missing any AMR genes that might be there, or prevent it, or not finding AMR genes that aren't actually AMR genes. So the whole problem of false negatives and false positives. And depending on if you're doing clinical work, you maybe don't want to detect some more questionable AMR genes and then find out later they weren't AMR genes. Whereas if you're doing more research-driven work, you might be interested in, I want to find anything that even looks like an AMR gene, and then I can do some more work on that later. So it does come down to those kind of issues. And also the other thing is what tools are associated with the database. And Dr. Feldgarden is going to talk next about that in a lot more detail. And I see there's a few questions along those territories of, what cutoffs, what thresholds are being used, but that will kind of jump into, that'll be part of that discussion. But there's a lot of difficult answers and questions in there. Are all the databases you've mentioned only for whole genome sequencing data sets?
No, so some of them, you can also use them. They have extensions to use for metagenomics, metagenomic sequencing, and I believe some are for reads as well, if you only have reads or don't want to assemble. And then I know that the hackathon that just happened are working on long read sequencing. So hopefully there's some tools coming for that soon too.
Yeah, and some tool like the MegaRes database, for example, is built around metagenomic reads, which we're not going to talk. Metagenomics is a whole other item that we might touch on a bit in the panel discussion, but has a whole bunch of other complications to deal with because obviously you're combining sequencing data from lots of different bacteria at once. So which resistance genes do you care about? How do you tell whether one is from a pathogen or not, for example, or is mobilized? Do I need to, it kind of relates to something we kind of already touched on. Do I need to validate in the lab predicted AMR phenotypes, or is the prediction on the genome enough to tell us about the AMR? You kind of touched on that.
Yeah, I mean, ideally, of course, if you could experimentally validate, that is, a really good way to know if what you're predicting is true. But it does give you a good idea of what is happening, and it depends on what you're using it for. So, for example, if you're using the output to treat a patient, I would definitely expect you to experimentally validate. But if you were just looking from a genomic surveillance point of view, then perhaps just predicting and giving a global overview is good enough.
Okay, one question. So for people that are new to the field, and you've given a great overview of the databases, okay, I want to use the CARD database. Where can I find some more kind of guidance to actually using the CARD database, like the nuts and bolts of, exactly how to navigate it and things like that?
Yeah, so specifically for the CARD database, there are a lot of tutorials that include videos as well as step-by-step guides. And in a lot of the resources, they have that as well. And so you can just go to like the main page and you should be able to find some help or step-by-step guide that will really help you through each of the resources themselves. And I also have links at the bottom of each slide that will sort of give you that information as well.
Grand. Like I know, for example, that, and it might be of interest to a few people here, that on Coursera, the ResFinder group at DTU have actually put together kind of a longer version of this course that kind of goes through the nuts and bolts of, you know, how to take your reads and end up with a like full sort of set of AMR gene predictions. So that might be a good resource, especially if you're new to the field and want to kind of do more AMR genomics, and if there's any questions that we haven't covered today. But yeah, for the most part, I would say the databases are all pretty keen for people to use it and pretty happy to help. I know they all have various resources tied to them, and I posted a link to the cards. Kind of, they do a kind of an annual state of the card kind of set of presentations that include sort of like in-depth videos of navigating the various bits of the website that Kara kind of highlighted. Okay. Who do you collaborate with, Dr. Tsang? Can you share your collaborators and where they are?
So I guess I just finished my PhD maybe like 3 months ago now. And so there I collaborated with Dr. Finley McGuire here. And we also collaborated with Dr. Rob Biko, also at Dalhousie, and Dr. Jerry Wright, who is at the Institute of Infectious Disease Research at McMaster University. And so they did a lot of the wet lab experimental work that I was talking about with the antibiotic resistance platform. And then currently in my role looking at Klebsiella genomic surveillance, we collaborate with the Institut Pasteur and the Center of Pathogen Genomic Surveillance, so CPSG, who runs Pathogen Watch.
Great. Okay. Which should come first to understand AMR, genomics or bench work? I know you did a bit of both.
Yeah. So I think this is kind of like a chicken or the egg problem. But I think it is iterative. And so I think like if you find a new resistance determinant through using genomes, you do have to experimentally validate. And then again, of course, if you do find like, if you do experimentally validate first, then it would be interesting to, you know, do genomic surveillance to see how widespread this resistance gene is. And so I think they sort of both go hand in hand, and they're both really important to studying AMR, essentially.
Mm-hmm. So there's several kind of questions specifics of AMR gene detection that I think will probably push on into the discussion at the end of Dr. Feldgarden's talk. And then we'll also kind of cover some of them in the panel discussion, because I think there's a lot of relevant factors. But here's a good and interesting one. Can there be genes which act as AMR genes and virulence factors? What is the difference between an AMR gene and a virulence factor? And I know Kara has spent a lot of time trying to develop ontologies to try and distinguish the two.
Yeah, so I'm not as familiar with virulence factors as other folks, I think, in the room, but there is a distinguishing factor between AMR genes and virulence factors. I'm not sure if one can act as both. Perhaps Dr. Feldgarden would answer that question better because his AMR Finder Plus does integrate finding both AMR genes, virulence factors, and stress response genes. So yeah, maybe I'll just divert that question to the expert.
But yeah, it's the classic problem in biology of, you know, where is the exact cutoff between these criteria is always the challenge. Do you know if there's any AMR gene resource, any resources that and databases that use secondary structure of proteins and incorporate that into their either database or then their underlying detection models?
There was one tool. I'll look for the paper and put it into the link after, into the chat box after, but I believe they did predict based off of sequence and then structure as well. I never actually used it, but I will put the link to the paper in the chat right after this.
Perfect. I think we'll go into the last question, but don't worry, keep asking questions. We will. Everything hopefully should be addressed in various discussions. how much resistance is explained by mutations versus presence or absence of resistance genes, such as in E. coli? And is there any database just for mutations? Or what databases would you use for mutations? I know you covered it briefly, but.
Yeah, so ResFinder has an additional tool called Point Finder, and they have a database just of chromosomal mutations for a specific number of species, and AMR Finder Plus in their NCBI reference catalog also have specific chromosomal mutations that you can pull from depending on the species that you're working with. In terms of the percentage between genes versus mutations, I think it depends on the species you're working with. So for example, in Mycobacterium tuberculosis, I think it's almost entirely based on mutations, whereas in something like Klebsiella or E. coli, it's more acquired gene based. And so I think it depends on the organism that you're studying.
Perfect. Okay. Yeah. Okay. Well, one last one. We got 10 minutes till 8 minutes till Dr. Felgarden starts. And I know that you took some of this route. So, as someone interested in AMR research, how and where would you advise to start, especially for someone whose background is in computing, or someone whose background is in biology? How do you make that transition into that kind of interdisciplinary bioinformatics area?
Yeah, so that was sort of how I started out as well. When I started in Dr. Andrew MacArthur's lab, I didn't even barely knew how to turn on my computer. So I think a good way to start is to, firstly, I think you can use the websites and the resources that are provided where you can just click around and insert a genome that way, just to get yourself familiar with the different nomenclature and the different I guess, words and context that is used within each of these databases and resources. And then once you sort of find yourself comfortable, or you have to do things more high throughput, meaning you have to analyze 100 genomes instead of one genome, then you can start pushing yourself towards the command line. And for that, I think I would definitely recommend just Googling some basic Linux tutorials just to get your feet wet in inputting commands and using the terminal. And then from then on, you can then go on to each databases and tools GitHub, which will provide you with how to install through the command line if you do want to do more high throughput analyses. And so that's sort of how it would get started first.
Perfect. Well, thank you very much, Dr. Tsang. I even see there's people asking for email to talk about Klebsiella and challenges of Klebsiella. I'll let you kind of navigate. And,
sounds great.
But yeah, great. And
thank you so much.
Dr. Tsang will be back at hopefully the end in the panel discussion to address some of the things that haven't been addressed once we've had a chance to kind of cover all the bits and pieces of this workshop, and everyone has a kind of see which questions are really crystallized. So there were some very common sort of themes and difficult, important themes that are when using genomics for AMR. Okay,